Good morning, dear friends. So myself, Dr. Chandrakant Shivaja here, MEG College, Malegaon Camp, Malegaon, Nasik. So today we start this last chapter, fifth chapter, that is pH meter. So in first sem we complete the practical based on these pH meters. So in first sem we have the practical in which we make different buffer solutions. We determine the buffer capacity. We determine or another experiment in which we can measure the pH of different solution, that is soft drinks, shampoos, soap solutions, etc. So we already use this pH meter in first sem practicals, but this chapter is in second semester in analytical chemistry. So we start this pH meter. What is the definition of pH? So this part we completed in practical course. The term pH was introduced by Sorensen in 1909, and it is defined as this pH is a negative logarithm of hydrogen ion concentration, H plus ion concentration, or it is a potency of this hydrogen ion concentration. And thus this pH of solution is the negative logarithm of hydrogen ion concentration expressed in molarity. This electrode in pH determination. This potentiometric method is most accurate method for determining the pH of solution. For finding the pH of solution, one should use an electrode that is a reversible to H plus ion. See, this reversible means this H plus ion can be adjusted. For this adjustment, if we go to the next slide, then we know this. So this electrode must be reversible. See, very important, what is this reversible to H plus ion? This H plus ion concentration is changing. This glass electrode, which is the reversible to H plus ion, is the most commonly used as an indicator electrode. See, in our practical we also use this glass electrode attached to this potentiometer. Generally this calomel electrode is used as a reference electrode. So in this potentiometer, pH meter, we have this reference electrode as a calomel electrode. So this calomel electrode and the glass electrodes are described below this. So what is this calomel electrode? It is a reference electrode. In this figure we have this structure of this calomel electrode. So this is a glass assembly in which, see, from bottom side we consider this. So this is the platinum wire, and this platinum wire have the connection between mercury, and it is connected to this external wire, that is a copper wire. So this mercury is filled in this tube. It is connected with this platinum wire, and this platinum wire is connected to this external circuit by using this copper wire. This mercury is in combination with mercury chloride. So this dark portion, black portion, is a combination of, it is a paste, and this paste is made from mercury and mercury chloride. Above this we have this solution. What is this solution? It is a KCl solution which is saturated. We know the meaning of saturated solution. We add more amount of KCl to make a saturated solution, and we have this side arm for this connection with another electrode in salt bridge. So this wire is used to measure the EMF, copper wire used for connection with this instrument, that is potentiometer. So this is the calomel electrode. So in exam there may be the question, write a note on calomel electrode or working of calomel electrode. So we have this diagram, and in this diagram we explain these different parts to explain this calomel electrode. Working we see in next slide. These calomel electrodes are very stable and good reversible electrode. They are extensively used as a secondary reference electrode. See this secondary word, it is a reference electrode, but it is a secondary. Which is the primary then? That we consider in next slide, that is the internal reference electrode. So this calomel electrode used as a secondary reference electrode. They are also easily prepared. A typical, just now you see in this diagram how we prepared, how we fill this calomel electrode. A typically of this electrode is shown in figure in previous side. This electrode is represented at that connection. Just now we see from bottom side, this copper wire is connected to this mercury is connected mercury, and this mercury chloride paste is there, which is connected to this saturated KCl solution. Generally this solution having the normality 1 or 0.1 normal solution. This electrode consists of a glass tube with a side arm. This pure mercury is placed at the bottom and it is then covered with a paste of mercury chloride and mercury. Just now we see in figure. This electrode is a saturated KCl solution. This concentration of solution may be 1 normal, 0.1 normal, or it is saturated solution at 25 degree. This saturated with mercury chloride. This copper wire, just now we see in the back side, this copper wire is introduced into the mercury for connecting the electrode to the external circuit. When we place this electrode in circuit, at that time we use this copper wire for connection. The other side tube is a lead into the salt bridge. This side tube, glass tube, is being inserted in the salt bridge coupled with the other half cell. The EMF of it depends on the process spontaneous with respect to the hydrogen electrode reaction. Next slide. We have this different type of calomel electrode. In previous figure we have this one electrode and another, this two diagram is there. So in this case also side arm is there, similar to this first diagram, but in this case we have the saturated solution, mercury solution, this calomel paste, that is mercury chloride solution, and for the bottom side we have this sinter glass plug. So this is the connection with another electrode. So side plug is there, this is the side arm is there, in this case we have the side plug is there. So this is the different form of calomel electrode, but assembly is same, this connection is same. This is the external wire, copper wire for connection coming from mercury. A number of form of electrode vessels suitable to potential use have been designed other than that shown in the previous figure. So in previous figure, and we have these two type of slide. Two more agar salt bridge type of probe type of shown in the above figure. These are the two more example. Vessels are changed but connection is same. Second type, that is the very important, that is the glass electrode. This is the actual working electrode. It is known as a indicator electrode. So it is a working electrode to determine the pH of solution. So it is also known as the indicator electrode. So what are the diagram? This is the diagram, different parts are there. So from bottom side we consider this is the pH sensitive glass. So this small portion of glass is very sensitive, very thin, fragile, and it shows the connection with our solution, our acidic solution or basic solution. So inside this bulb we have this internal reference electrode, that is the AG and AgCl. This is the internal reference electrode, silver in connection with silver chloride. So this is the primary electrode, that is the internal reference electrode, and this calomel is a secondary electrode. About to this we have this internal filling solution, that is 0.1 molar HCl solution is filled in this glass. See, this round bulb is filled with the 0.1 molar HCl solution. In this solution we have this internal reference electrode as a silver chloride, and this internal reference electrode is connected to the mercury. This mercury have the connection with lead wire or copper wire. So in this case we have this lead wire. What is this? This is the high sensitive glass, outer glass. Inside this what we have, this resin filling. This is the resin filling support for this. Outside this what we have, this metal cap. This metal cap is there. In this metal cap we have this tube which is inserted by lead wire. विद्यार्थी मित्रांनो, दिस हा ग्लास इलेक्ट्रोड खूप इम्पॉर्टंट आहे पीएच मीटरमध्ये. दिस स्मॉल पोर्शन इज व्हेरी सेन्सिटिव्ह फॉर pH, and by changing, see, we have this reversible. So this glass layer, this having the deposition of H plus ion, outside we have this deposition of H plus ion, we have this EMF. So it is a reversible. When we dip in distilled water, this layer is vanish, outer layer is wash out, and thus this glass electrode used for number of times. So it is a reversible electrode, very important, reversible electrode and indicator electrode, which is used in actual pH determination. So what is the working? See the next slide. When a glass surface is kept in contact with a solution of potential is developed between the glass and the solution. This potential thus established as a function of H plus ion concentration of a solution and the nature of glass electrode. This EMF, this potential develop, it depends upon the H plus ion concentration or the glass electrode type. The most widely used hydrogen ion responsive electrode. Although there are other more conventional electrode system for measuring pH, the glass electrode becomes it a convenience, is used most universally for pH determination today. So this glass electrode is a most convenient electrode for pH determination. A typical construction of pH glass electrode is shown in previous slide just now. So this glass electrode is a actual working electrode or an indicator electrode. What are the advantages of this glass electrode? First, this electrode is used in presence of oxidizing and reducing solutions. So we can determine pH of oxidizing and reducing solution. It is also used to measure the pH of very small solutions. If you have the small solution, then that sufficient amount of solution, that small amount of solution is sufficient for determining the pH. Third, this solution, this pH glass electrode is used to determining the pH value from 1 to 12, and in some special cases this glass electrode is modified, and at that time we can measure the pH up to 14. This pH 14 means towards the alkaline solution, more basic side. So we have the more basic solution, at that time this glass is attacked with that base or glass is, so that we have this special glass electrode to measure this high basic solution, that is pH 14. So general glass electrode we measure the pH up to 1 to 14, 1 to 12, and special glass gives measurement of pH up to 14. Next, this equilibrium between the outer and inner solution of the glass membrane reaction is reached quickly. So just now we say that quick small portion in which inner solution, that is 0.1 molar HCl solution, and that outer solution, that is a test solution, that H plus ion layer formation, membrane formation outside and inside of this glass, sensitive glass, is reached very quickly. That is very advantageous of this glass electrode, and we give this potential, we develop that potential, and we read the pH of solution. So it is very simple to operate and free from poisoning. It is also used for automatic recording and control of industrial and commercial processes. It is also used in colored solution, colloidal, turbid solution. जेव्हा आपलं सोल्युशन कलर्ड सोल्युशन असेल, कोलायडल पार्टिकल असेल सोल्युशनमध्ये, टर्बिडिटी असेल, तरी त्या सोल्युशनमध्ये वी कॅन डिटरमाइन दिस pH by using this glass electrode. So very important, these are the advantages of this glass electrode, but till it having some disadvantages. What are the disadvantages of glass electrode? See one by one. This glass electrode is made of a thin glass. Just now we see this thin glass is there, very thin, and this fragility, it must be handled very carefully. ग्लास असल्यामुळे, ठिसूळ असल्यामुळे इट इज हँडल करताना खूप काळजी घ्यावी लागते. हा जर बल्ब उघडा असेल आणि आपण बिकरमध्ये टच केलं तर ग्लास फुटू शकते. सो इट इज हँडल विथ व्हेरी केअरफुली. सो इट हॅविंग अ कवरिंग ऑफ प्लास्टिक round, so it is circulated with this plastic cover or plastic cover instrument is there, inside that this bulb is placed. So instead of glass, this plastic is touched to this beaker. The glass electrode, we see this glass electrode in practical use. This glass electrode membrane has a high sense resistance, and so this resistance is very high. Glass having a high resistance, so ordinary potentiometer cannot be used for measurement of EMF, and thus this electrode potential is measured by using specially prepared potentiometer. So that is another advantages. Third, this glass electrode must be rinsed thoroughly with water and then standardized before use. See, these layers are formed inside and above this glass, and thus it is rinsed with distilled water before use to wash out that outer H plus ion layer. So that is also disadvantage. Before use we should wash it. Actually it is a normal process. Before use that glass electrode it must be washed with distilled water. It loses its sensitivity when dry or during storage. When this glass electrode keep for long time, at that time this is drying, that solution 0.1 molar solution HCl solution is there, so drying process is occur, or it must therefore be kept sensitive by immersing it in distilled water. For absorption of water we place this glass electrode in a distilled water, and for at least 12 hours prior to use. So generally this is also disadvantage. Generally we place this glass electrode in distilled water for storage also. आपल्या लॅबमध्ये आपण जर पाहिलं ग्लास इलेक्ट्रोड आपण डिस्टिल वॉटरमध्ये डीप करूनच ठेवतो. So that disadvantages is automatically removed. Or if we stored it disconnected or without water, then before use you should immerse that glass electrode in distilled water before 24 hours prior to use. वापरण्यापूर्वी 12 तास अगोदर तो पाण्यात भिजून ठेवा, नंतर वापरायचं. आपण ही काळजी लॅबमध्ये घेतच असतो. वी स्टोर दॅट ग्लास इलेक्ट्रोड इन डिस्टिल वॉटर ऑल्सो, डिस्टिल वॉटर. नेक्स्ट, it cannot be used in pure ethyl alcohol, acetic acid and gelation. It cannot be used in strongly alkaline solution. It cannot be used at high temperature above 100 degrees centigrade for a prolonged period. So these are the disadvantages of glass electrode. Till it is very useful. This pH meter, the most sensitive electrode, which consists of thin membrane of glass, which is responsible to changes in H plus ion concentration, is used to measure the pH of solution, and the instrument is referred to as a pH meter. So what is pH meter? So we have the sensitive electrode. In this case we have this glass electrode. This H plus ion membrane is formed, and this difference layer is formed due to concentration, various concentration of H plus ion, and that different layer gives the EMF, and we measure that EMF as a pH of that solution in pH meter. This potential of the glass electrode for the two solutions separated by a glass membrane. So there are two solutions, internal solution that is 0.1 molar HCl, and this external solution as a test solution. And this glass membrane having that two solution, and it gives this EMF is produced, this potential is produced by using this Nernst equation. What is this? This EG, EMF of glass electrode, G for glass electrode, is equal to RT. R is the constant, T is temperature, F ln of concentration of H plus ion 1, concentration of H plus ion 2, that two solution having two different concentrations, H1 concentration of first solution, concentration of second solution, is equal with 0.059 into bracket pH of second solution, pH minus pH of first solution at 25 degrees centigrade. So this gives the EMF of glass electrode. The glass electrode has high resistance, 1 to 100 mega ohms. This resistance is 1 to 100 mega ohms, and therefore this instrument were specially designed to meet the requirement of the glass electrode. These instruments are called as a pH meter. This pH meter are of two types. First is potentiometric or slide wire type, and the second is a direct reading type. So what is this potentiometric or side wire? This potentiometric or side wire type pH meter, in this type of instrument, glass electrode is incorporated in an ordinary potentiometric circuit. So this is the glass electrode, see here, this cell with a glass electrode. So this is the cell in which we have this glass electrode, and it is connected with a circuit. So these are the different circuit. Off balance currents are amplified electronically so that millimeters can be used to detect the balance point. The electronic amplifier is this in this manner act as a null point detector. A side wire is adjusted until no meter deflection is observed. So this is the meter in which we get the deflection. So initially we get the null point, 0 deflection, and then after dipping this glass electrode we get the deflection, that is we measure the EMF. A side wire is adjusted until no meter deflection is observed. This meter shows no deflection, 0 deflection. The side wire is calibrated directly in pH unit and in millivolts. So in this case we have this cell in which we have this glass electrode which is connected with resistance. So this ABC line is there, there is also resistance. This resistance is connected to this RC. So this potentiometer PC, it adjusted this side wire. In this we have these two connections. This Weston cell is there, we introduce Weston cell, and for this adjustment of resistance of Weston cell we have this resistance R. This Weston cell is introduced by connecting with point 1, and this Weston cell is adjusted by using this R resistance. Then we connect this second point, that is the actual electrode or cell, and in this case we measure this deflection. For adjustment of temperature we have this resistance RT. So this is the parallel to this ABC resistance, and this RT resistance is adjusted for the temperature change, and by using this resistance we can measure the pH in this detector, that is a pH meter. So this is a meter which shows the deflections. So this is the type of pH meter in which this potentiometer or side wire, these are the side wires, is used for adjusting the resistance. So this is the point in which we standardize this. We have this equal point in which we standard our cell, that is potentiometer. Second type is a direct reading type, and this type of instrument we use in practical laboratory. Direct type of pH reading. The following few arrangements in the direct reading type of pH meters and their function should be always remember. What are these points? There are 3 points. First point, that is a control knob, temperature knob, and the third point is a slope control knob. So that point we have to consider in our next lecture. So till today we have completed the topic, that is a pH meter. What is pH? What are the electrode, reference electrode, glass electrode, advantages of glass electrode, disadvantages of glass electrode. And this, for measuring of pH we have this 2 type of potentiometer, that is potentiometric or side wire, and the second is a direct reading type of electrode. So this direct second type we consider in our next lecture. Till then, thank you.